Hi, I'm Ellis.
I'm Ava. And I'm Lila.
This is our video project on the PDGF signaling pathway related to blood clotting after injury. Our video is 212. This is the PDGF. PDGF promotes the growth and division of various cell types. This is crucial for tissue repair, wound healing, and normal development. General information. The signaling molecule of PDGF is primarily hydrophilic and it's located on the surface of the cell. PDGF is a stimulator of cell division in various cells. Some advantages of the PDGF signaling pathway are accelerating tissue repair, promoting collagen production, supporting blood vessel stabilization, and enhancing cell survival in neural and cardiac tissues. However, because growth factors stimulate cell activity, PDGF could potentially contribute to abnormal or uncontrolled cell growth. This could promote the development of several serious diseases, including cancer, which is a major disadvantage. Now, how does it work? This group of receptor proteins works by stimulating cell division. When someone is injured, cells called platelets are released from megakaryocytes and are sent to the injury site. These platelets release PDGF molecules that bind to receptors on target cells. These receptors are activated when the molecules bind to it, and they're able to use phosphorylation to bind phosphates to tyrosine receptors in an intercellular space and make ATP into ADP. This begins the signal.
Specific example. Imagine someone has a stroke. Well, PDGF has a role in strokes, where it acts as both a damager and repairer. There are two different types of strokes, ischemic and hemorrhagic. We will focus on an ischemic stroke. An ischemic stroke is when a blood vessel in the brain is blocked by a clot, cutting off oxygen and nutrients, causing rapid brain cell damage. It starts with damage to the blood-brain barrier. Platelets are then released in the injured area. This is what a normal blood-brain barrier looks like, compared to the blood-brain barrier after an ischemic stroke. There are four different types of PDGF signals used in repairing a stroke: PDGF-CC, PDGF-A, PDGF-BB, and PDGF-D. There are two different receptors for the PDGF, PDGFR alpha and PDGFR beta. There are many molecules used in the intercellular response of the cell. Some protect against apoptosis, which is neurodegeneration. Others are used for rebuilding vessels, cell growth, and cell division. The three main cells involved in repairing the aftermath of a stroke are endothelial cells, fibroblasts, and pericytes. Following an ischemic stroke, PDGFR beta expression in pericytes is upregulated. Upregulated means increasing the number or activity of receptors. This increases gene expression. This upregulation plays a vital role in regulating their survival, migration, and interactions with endothelial cells in the brain. PDGFB, produced by endothelial cells involved in blood vessel formation, activates PDGFR beta signaling to recruit pericytes and ensure the stability of newly formed blood vessels. PDGFR beta is crucial for repair, and angiogenic endothelial cells release PDGFB, which activates PDGFR beta to recruit pericytes back to the newly formed vessels, aiding in stabilization. Following the acute phase where the BBB is damaged, the subacute phase features increased production of PDGFC and PDGFD within the injured brain tissue. PDGFC and D ligands bind to PDGFR-A alpha and PDGFR beta on target cells, including fibroblasts, pericytes, and endothelial cells. Activated cells undergo proliferation, migration, and differentiation, enhancing tissue repair and angiogenesis. So how is the signaling pathway terminated? Well, the activated PDGF receptors are internalized into endosomes and subsequently sent for lysosomal protosomal degradation, which removes them from the cell surface. Agonist or antagonist signaling pathway. The antagonist is imatinib, which is a tyrosine kinase inhibitor, or TKI. It works by blocking specific proteins responsible for cancer cell growth and survival. It is exogenous. The imatinib effectively blocks the ligand-induced phosphorylation of PDGF by acting as a competitive inhibitor that binds to the ATP binding pocket and prevents the response from taking place. By inhibiting the receptor, it disrupts downstream signaling cascades. This changes the response because it prevents the phosphorylation, reducing cell proliferation and decreasing migration/invasion of fibroblasts and tumor cells. Thank you for listening.